followed by the plant introduction. Next topic is milestone in plant tissue culture. What are the milestone? The milestone is nothing but history of plant tissue culture. What are the history that can be how the plant tissue culture that can be able to develop? First scientist Haberland in the year 1902. He was the father of plant tissue culture. Why we call it as a father of plant tissue culture means already we know. He proposed the concept of totipotency. Here, cultural plant cell in an artificial, that is a laboratory condition, in a in vitro condition, under a glass, inside a glass, with the help of cultural medium. That cultural medium is nothing but a MS medium, white medium, Murasic and Scoob. MS medium is nothing but a Murasic and Scoob medium, white medium, B5 medium, etc. That medium, they contain the knob salt solution. That knob salt solution containing glucose and peptone in a particular proportion. That not only for the glucose and peptone, here they contain the calcium nitrate. The knob salt solution, they contain calcium nitrate 3.0 gram, potassium nitrate 1.0 gram, sucrose 50.0 gram, magnesium sulfate 1.0 gram, dibasic potassium phosphate 1.0 gram, and deionized water 1000 ml, that is a 1 liter. These contain the knob salt solution. Then it contains the glucose as well as peptone for the development of callus. What is callus? The callus is nothing but unorganized and undifferentiated mass of cell, that is called it as callus. Here, unorganized growth of cell and tissue. That was proposed by the scientist Haberland. Here, the totipotency means, simply we say, the development of entire plant from a particular isolated cell or a tissue in a laboratory condition. This is the first history about the plant tissue culture in the year 1902. Next scientist P.R. White in the year 1934. P.R. White proposed one of the white medium. He used that medium for the development of the root culture using the knob salt solution. But in case of white medium, he added 3 important vitamins like pyridoxine, thymine and nicotic acid. This is the one of the milestone for the plant tissue culture. Next scientist was F.C. Stewart. The F.C. Stewart, he proposed the coconut water as the medium, because the F.V. Stewart in the year 1948, he used coconut water in plant tissue culture technique or a work, because the coconut water is nothing but a cellular endosperm. That cellular endosperm that can be able to added in the plant tissue culture. So this technique is called it as a cellular totipotency, because that cellular totipotency that can be able to useful for the culturing of the carrot for as a explant in the year 1948. Next scientist Morrell and Martin et al. In the year 1952 and 1955. In the year 1952 and 1955. Both of the Martin and Morrell developed virus-free plant in one of the plant, that is called it as dahlia and potato plant, by using shoot meristem culture. What is the meaning of shoot meristem culture? In the tip portion, the meristem that can be able to present. In the some of the branches, lateral meristem, intercalary meristem that can be able to present. Here lateral meristem also present in the plants. That is call it as a shoot meristem culture that was developed by the Marl and Martin in the year 1955. Next scientist was Murasik and Skoog in the year 1962. Murasik and Skoog proposed MS medium, that is called it as a Murasik and Skoog. That MS medium mainly used for the plant tissue culture because the MS medium they contain the high carbon content in the medium. Then only it is the basic medium for the plant tissue culture work. For all the tissue culture work, it is the most frequent and suitable medium. Then next scientist was Cantel et al. In the year 1962. He produced and his worker produced test tube fertilization in flowering plants. You know what is fertilization? The fertilization is one of the process fusion of male and female gamete in test tube, especially in a flowering plant. Then next scientist was Yamada et al. In the year 1963. He produced callus, that is called it as cali. That cali that can be produced from free cells in tissue culture of one of the plant, that is called it as Tradescantia refluxa. That Tradescantia refluxa is one of the plant